According to the World Health Organization, more than 300 million people worldwide suffer from depression, and an even larger number of people struggle with anxiety. So it comes as no surprise that a lot of scientists are trying to find treatments for mental disorders like these. The most well-studied methods we have for treating them involve tweaking the level of brain chemicals, like dopamine or serotonin. We know that this strategy often works, and that many patients do feel better when given these kinds of medications. But they don't work for everyone, and they can have some side effects that patients aren't thrilled about, which is why one researcher is looking at the issue from a totally new perspective. Instead of chemicals, Kafui Dzirasa is interested in the brain's electrical activity. Now, lots of neuroscientists have studied the brain's electrical activity, but up until now, there have been limited ways to do it. One is to take an image of the whole brain at a particular moment in time, like with an MRI brain scan. Another is to measure electrical activity over a longer time using a few electrodes poked into a tiny region of neurons. But Dzirasa began his career as an engineer, so he wants to know how the whole system works, not just little pieces. He studies the patterns of electrical signaling across the entire brain over long stretches of time. And the discoveries he and his team are making could totally rewire mental illness treatment as we know it. Which sounds like the kind of story it would be cool to tell in person. That's why we created SciShow field trips, to see cutting-edge research in action. We sent our friend Madelyn Leembruggen to Dzirasa's lab at Duke University to learn how he is trying to engineer a better future for people with psychiatric disorders.
Kafui Dzirasa's title is almost as complex as the subject he studies.
My name is Kafui Dzirasa, and I am both an investigator with the Howard Hughes Medical Institute and an endowed professor at Duke University. But my favorite title is Vision and Value Scientist. I am also in the Department of Psychiatry and Behavioral Sciences. I'm in the Department of Biomedical Engineering. I'm in the Department of Neurobiology, and in the Department of Neurosurgery.
You sound like you get a lot of emails.
Yes, a lot of emails.
Just a little busy.
Yeah.
But the story of how he got interested in the brain is a lot simpler.
I grew up watching Star Wars. And there's this magical scene. Luke Skywalker has gone off, and he's fighting this supervillain, Darth Vader. For those of you who haven't seen it, I don't want to give it away. Turns out he's his dad, right?
Whoa. Spoiler alert. Now, I'm sure that a lot of future scientists who watched the Star Wars movies would have been inspired to try to invent a lightsaber. But Dzirasa was more interested in something else.
There's this scene where Luke Skywalker gets his arm cut off, and at the end, he's in this medical bay, and they've attached sort of this robotic prosthetic arm, and Luke Skywalker's doing this, and he's moving it again. And I thought, wow, wouldn't it be so cool to take engineering and science and figure out how to create body parts for people who are suffering, unable to walk or unable to move.
Dzirasa studied chemical engineering in college and eventually got interested in biomedical engineering, a field that combined healing and medicine with cutting-edge tech. But for Dzirasa, becoming the best biomedical engineer he could meant understanding the body inside and out. And that meant becoming a medical doctor, too. He went into medical school with the dream of creating robotic arms, but ended up focusing on a very different part of the body. During one of Dzirasa's first clinical rotations, he was introduced to a patient in the psychiatric ward, a veteran with schizophrenia.
And I walked in and I just asked him, how you doing? What brings you in? Are you having any difficulties? And he starts telling me about how he's having this headache. And filling out my form, and finally I ask him when the headache started. And he tells me about how he was in Vietnam and how he was captured, and how the people who captured him started drilling into his head. And ultimately the story ends with him telling me the people who captured him implanted a chip in his head. And my best guess at the time was that that is what we would call a delusion. I actually got really curious about how his brain had created this experience that probably didn't match all of reality, right? And how were the electrical signals in his head causing him to think differently?
But while the other doctors knew which medicines tended to work for illnesses like schizophrenia and bipolar disorder, nobody had a solid answer for what exactly was going wrong in this patient's brain, nor the exact solution needed to fix it. It was then that Dzirasa had a thought that would shape the rest of his career. If we can engineer a robotic tool that restores function to someone's arm, why can't we do the same with our brains?
I'm still an engineer, It's still about the brain. It's still about electrical process, and it's still about prosthetic devices. It's just now, instead of thinking about the parts of the brain that create motor function, you might be thinking about other parts of the brain, things that you'd hear about like the amygdala or other parts of the brain that shape emotions, and creating prosthetic devices for that.
And because the animating force behind both robotics and the brain is electricity, that's where he decided to focus his efforts. Dzirasa likes to compare the brain to a complex highway system, with cars as the electrical signals driving on roads, crisscrossing everywhere. So how does information travel in the brain?
Yeah, so for humans, for example, we have over 200 billion cells in the brain. It's a ton. And half of those cells actually can create and move electricity. When those cells create and move electricity, you can send those electricity down highways, right? Down highways and tunnels. And in neuroscience, we tend to call those axons, but they're basically highways and tunnels for electricity to move.
The cars are individual electrical signals, and they move through our brains via neurons, the cells in our brains that transmit information. There are little gaps between each neuron called synapses, and for the electrical signal to jump the gap, it needs a chemical called a neurotransmitter to pass it along. Those are the things like dopamine, serotonin, and noradrenaline. For your brain to pass a message along, the neuron releases a bunch of the neurotransmitter into the open space. It binds to the next neuron, which triggers the electrical signal. Then, once the message gets sent, those neurotransmitters unbind from the receptors, get released back into the open space, and then most are reabsorbed by the original neuron to be used again. And while this is a nice, tidy system in lots of people, people who struggle with mental illnesses often don't have enough of the neurotransmitters they need to keep the right parts of the brain actively signaling. So what kind of treatments for psychiatric disorders currently exist, and what are the benefits and downsides that you're trying to maintain or to solve for?
Brain cells release chemicals, which we call neurotransmitters, and those neurotransmitters convey information to other cells. So that is what people are typically targeting when they say brain chemistry, right? You're targeting either the synthesis or creation of these chemicals, the release of these chemicals, or the ability of these chemicals to connect with the other cells. And many of our medications do that. The greatest challenge with this class of chemistries is that the medication you take goes everywhere. And so it can have effects on cells that you don't want to be targeted, right? And so some of these same sort of connections or receptors, things that connect to the chemistry, is in other parts of your body. It's why you might take a medication for some form of psychiatric illness, which may make you nauseous, right? Because those same sort of connections, those same sort of cells that produce electricity are also in your gut.
Which is why Dzirasa thinks that looking at mental illness in a new way could open up treatment opportunities for the people that can't be helped by what's currently out there. And it starts by considering the entire brain. If you really want to understand the whole brain, you have to look at what's happening with the flow of those electrical signals across regions, and not just the chemicals in the synapses.
If you want to make sense of all of those cars and where they're going, you ultimately need something like Google Maps or Waze, right? Because there's constant movement that you have to understand how all of those pieces are working together. That's exactly why we run into a problem, right? As soon as you start thinking about how many cars there are on the road, you now need to make sense of the patterns, what's changing from moment to moment and day to day.
So Dzirasa started studying the patterns of electrical activity that underlie emotions, as well as what those patterns look like in people with psychiatric disorders, or even people who are under a lot of stress. We have this kind of electrical map for other parts of the body, by the way. When a doctor wants to check how your heart is working, they measure the electrical activity of your whole heart using something called an EKG. Right, because I've had like an EKG done before, and that in itself seems complicated, but then you talk about the size of the brain, so it's much, much harder to map something like the brain than it is the heart.
That's right. I came to appreciate this. I'm also a physician. And so I've also put those 12 leads of EKGs onto somebody's chest to measure information, electrical information in the heart. And from those, what we call 12 leads, or sort of 12, stickers that you put on, you can see 12 electrical waves. So for each of those leads we put in the brain, we also get a wave. But in this case, we're not getting 12 waves, we're getting 1,000 waves. And so you can see how quickly this becomes a challenge, right? You're getting 1,000 waves, but then you're also getting another 2,000, 3,000 individual brain cells in their activity. And then you have to find patterns in all of that. We want to find the patterns that show up over and over again in this large electrical data set that includes brain waves, like what you would get from EKG. The data set and the scope of the brain, it's so much more complicated. Electricity doesn't just flow in this direction, it flows in every direction.
When Dzirasa was a postdoc researcher, he decided he wanted to map how electrical activity in a mouse brain changes across time. And he needed some very specific tech to be able to do it. So Dzirasa invented it himself. It was the first example of Dzirasa using an engineering approach on a biological question. We also talked to lab manager Stephen McGew, who walked us through some of the details.
So this is how we build our electrodes in-house, based on an approach that Kaf developed when he was in grad school. So we start off with a single wire, as you can see.
Barely see it.
No, that makes sense. It's only 50 microns in diameter. And so this is a tungsten wire that is insulated throughout so that only the exposed tip, which will be implanted into the brain, can record electricity and pass it along to the other end, which will be attached to the other parts of the electrode. And so we sort of gently put it into this grid, and this allows us to tailor each electrode to the specific sort of conformation of the brain region that we want to target.
And so we can send those electrical pulses from one brain area down to the other. If you look here on the screen, you can actually see those electrical pulses. You can see them and you can hear them. And they sound like tiny crackles and pops in the background. This is 4 different brain cells out of that 100 billion that can create electricity. That is the talking of brain cells, sending that electrical information down the highways and tunnels to other brain areas.
His ultimate goal is to create an electome, which is like a genome, but for the human brain. Can you tell me about the electome?
Yeah, so we came up with an idea. It was, 2015 or 2016. And the idea was, having gone into the clinic and seeing what an EKG can do, you could see the brain in action. And from these patterns of these 12 leads, you can determine what was going on with the heart. We wondered if you could do the same type of thing with the brain. And so our idea was, well, maybe the brain generates emotions in the same way by coordinating large parts of electrical activity together. And could we find patterns, which will say patterns of functional activity or electrical activity? And we ultimately called that the electome, electrical functional connectome. And so you might have a pattern that shows up when you're happy, or a pattern that shows up when you're sad, or a pattern that shows up where you really want to hang out with your friends, or a pattern that shows up when you might be a little bit anxious or scared. And so that was the idea. Could we discover these patterns in preclinical models and then see if these ultimately showed up in patients down the line as a diagnostic? We as neuroscientists have been pursuing this challenge for some time, and we got really excited as a nation on this effort in around 2013. There's a huge initiative launched called the Brain Initiative. It's a great name for understanding the brain, right? And the goal was to create a new class of technologies to make sense of the brain. We realized at the time we were recording, 20 or 30 brain cells at a time in preclinical models, and we needed to get up into the tens of thousands or hundreds of thousands. Well, the brain is electricity, but it's also chemistry. And so we also needed to create new sensors for brain chemistry and brain electricity at the same time. And then you needed machine learning or AI-based tools to integrate all that information together. And then that tells you sort of about physiology. It's a massive undertaking, but we're really optimistic about what this will tell us about the brain and ultimately how changes in the brain produce illness so that we can come up with treatments and cures.
Step 1, build the map. Step 2, identify the roadblocks. Step 3, figure out the detours. It's an exciting trajectory with a lot of potential. even while we're still at step 1. And one of their first big successes in building up these neurological highway maps came when they identified the electrical signal network underlying stress resilience in mice. Stress can work as a kind of switch that might turn on any number of psychiatric disorders in certain people.
When I was training as a psychiatrist, I would go in the hospital and I would see different patients. I kept seeing the same thing over and over. And that was when they would end up in the inpatient psychiatric unit, many of them had a major stressful event a little bit before, right? In some cases, somebody was having problems with a family member. In some case, the major stressor was just like the student going off to college for the first time and missing home, the stress of exams. So what I ultimately came to realize was that if I understood how stress triggered all of these things, maybe I could come up with a brain pacemaker that helped with stress. And if you do something like that, maybe you can help everybody who might have problems down the line before the problems start.
And they're not just focused on the downers. Long term, they also want to understand other feelings or experiences, like love, pleasure, and even hunger. He also wants to use those findings to correct the patterns when they're causing the symptoms of mental illnesses or neuropsychological disorders. Dzirasa's team is investigating a group of proteins called connexin proteins, which sit at the end of neurons. Sticking with the whole roads analogy, you could think of these proteins as two halves of a drawbridge. The cars only get through when both halves are connected. Not enough connections, and you get these neurological traffic jams, or parts of the brain that should communicate but don't. So if you need more cars to get through faster, one way to do that might be building more bridges. And in this case, they want to add more connexin proteins to see if that makes signals travel better across neurons. But not in humans, or even in mice. This time, they wanted to look at worms.
I am Julia Dirk, and I'm a senior scientist for Howard Hughes Medical Institute, and I study worms.
So why are C. elegans the right worm to use for this kind of study?
Yeah, so any invertebrate would be a clean system because they express innexins, not connexins. So as opposed to when we put the connexins into the mouse brain, we have to worry about them potentially binding to something else in another connexin. Like their connexin 36 is all over lots of neurons, but the worms don't have that. And so it's a really great way of understanding that the manipulation is only really happening in that particular neuron subset and with those particular proteins.
They're basically adding in synthetic bridges to see if the electrical signals would use them too. It's a specially engineered form of a connexin protein called LINX. LINX stands for long-term integration of circuits using connexin, which is a mouthful, so LINX for short.
What's really wild is that we've now, through mutagenesis, of changing one amino acid at a time, changed these connexin proteins that are usually found in a white perch fish. And so they should only bind to each other. And that's really a huge component of what makes LINX special, is that it's not any of the connexins that were found out in the wild. It's slightly modified connexin proteins that should only specifically dock to and work with and bind to each other.
But then they tried the DIY connexin proteins on mice, which was trickier because they're mammals, and mammals do have their own connexin proteins. That means there's the potential for the proteins to link up with the ones that are already there and totally mess up the signaling. One errant protein might result in your fancy bridge getting built to nowhere. To overcome that problem, more engineering.
So we use viruses that are target specific brain regions or different cell types. And so, for instance, a virus essentially is just a replication machine, but we can sort of remove the parts of a virus that we don't want replicated, but use the inherent machinery that basically replicates its own genome, and then we can splice in the parts that we want to have sort of made for us.
So you kind of scoop out the virus stuff inside the virus and use it as a little delivery vehicle.
Exactly.
So what part of the brain are we looking at right now, and what is it responsible for?
This part of the brain is called the striatum, or nucleus accumbens, and it plays a really big role in how the brain experiences reward. So in the case of illness like depression, the activity in this part of the brain might be altered or changed or decreased.
So is the red that's showing up there the connexin protein?
That's exactly right. What we're trying to do is see how well the connexin proteins are expressing in these brains. The idea is in the future, we will use these connexin proteins as a potential treatment. So we want to make sure that we can get them in the brain and that they change the way the brain is functioning.
So the goal with all of this is to find treatments for mental health disorders that work for the people that can't take traditional medications, and have another tool in the tool belt when it comes to treatment-resistant disorders. What would a treatment based on connexin protein look like in a human eventually? Maybe.
If you want to get certain proteins into the body, it's pretty easy, right? You can either ingest it, or if you go to the doctor and you have a syringe, you can get an injection. Now, getting them in the brain is a little more complicated because your brain has a lining that prevents things from getting in. And as part of the Brain Initiative, they were creating tools to overcome that challenge of getting things in the brain. One used this type of technology where they created nanoparticles, these really small particles in which you can put things inside. And you could use a device to open up this barrier just slightly so the nanoparticles could go through. This device uses sound energy. And so you basically put sound waves in the brain. They gently shake the blood-brain barrier, it opens up and the nanoparticles go through. It's called focused ultrasound. If we get our connexin proteins inside these nanoparticles, we could open up the blood-brain barrier and it'll carry them into the brain. The second type of tool that the field was creating was called adeno-associated viruses, or AAV viruses. So the virus is sort of the delivery mechanism. So down the line, that's sort of the treatment that we could anticipate. Someone would go into the doctor's office, and in the doctor's office, they might one time in life open up the blood-brain barrier or give them this essentially vaccine which carries the connexin proteins to the right location in the brain, forms these connections, and then makes individuals more resilient to the impacts of stress.
Dzirasa's work is pushing the boundaries of how we think about mental illness and psychiatric care. These innovations could be the start of some truly phenomenal breakthroughs and help a lot of people who need it. The road may be long, but at least we have people like Dzirasa building bridges.
SciShow field trips are made with our friends at HHMI Tangled Bank Studios. We've come together to bring you face-to-face with researchers at the cutting edge of scientific discovery. You can watch more of Tangled Bank's science content at tangledbankstudios.org.